name is Chris Rhodes, and I'm standing in for my colleague Lakshma Reddy, who is a scientific director at Drug Delivery Experts and put these slides together for me. I'm a chemist by training with a Ph.D. at UCLA and postdoc at Yale University with over 25 years formulation and drug delivery technology, development, and clinical translation experience. At Drug Delivery Experts, we work on all sorts of injectable systems, from solution to complex formulation and small molecule to antibody modality. We have extensive experience in long-acting systems, from microparticles to gel-forming systems and injectable suspensions. In this presentation, we're going to focus on milling techniques at the small scale for proof of concept testing for nano and microparticle suspension formulations. As is well understood, smaller particle size increases surface area and increases the rate of dissolution of drug. This has been well established in oral and inhalation products. In the case of long-acting injectable suspensions, particle size reduction serves to increase the dissolution rate for very insoluble drugs and to allow for injection through reasonable needle sizes for patient convenience. The control of particle size also allows for higher concentration suspension formulations to be feasible with reduced injection volumes. Some of the concentrations required for the long-acting systems become quite significant. Various micronization techniques are used for milling crystalline drug formulations. These include ball milling, microfluidization, and jet milling. One important note is that for these techniques to work successfully, the drug must be crystalline with a melting point over 150 Celsius or so, and have very low solubilities in the vehicles used for injection, typically aqueous or oil-based systems. The techniques we use in our lab at the small scale consist of ball milling and vary from a low-energy roller mill to a medium energy vibratory mill to a high-energy planetary ball mill. Usually, inert beads of various composition and sizes are used in these mills to generate additional energy for grinding crystalline materials. Here's a picture of a roller ball mill, which provides very low shear force and can be conducted in plastic vials or centrifuge tubes at very low drug mass. The low energy is sometimes capable of reducing particle size to the 5 to 10 micron range. Particle size will be dependent on the bead size used, as well as the quantity of the beads relative to the drug and the suspension concentration. This technique is usually done wet, where drug is turning and beads are turning over each other continuously, as shown in the picture, to create a grinding motion. The benchtop mill or ball mill provides more force and uses a vertical or horizontal motion to create energy with beads and drug contained within the jars. The technique can be done at a reasonably small scale and is typically done as a dry powder. These instruments come in various sizes, with the smallest containers of about 20 milliliters in size. This is a fairly high energy milling. Planetary ball mills come in various sizes and can be operated at medium to high energy. The importance of these is that their mixing is quite efficient because of the dual planetary and ball grinding jar motion. The smallest bottles are typically 100 milliliter jars, and they are made of stainless steel or ceramic. Technique is usually completed as a wet suspension, and one advantage of this technique is that it can be done at very high concentrations because of the multiple motions and efficient mixing. Microfluidizers have been used and were in fact first popularized by Elan Nanosystems to make particulate suspensions for oral and injection use. Pressure from a piston is used to create particle size reduction of drug in a suspending vehicle. One limitation of this technique is that the concentrations in the system are relatively low, and many passes are required through the chamber to achieve particle size reduction. Typically, formulations from a microfluidizer need to be concentrated by centrifugation because of this low concentration. Jet mills are high energy mills and are used as dry powder grinders for drug. We're not going to spend any time on this system today. There are a number of parameters that are important in the control of particle size produced in the milling systems. First and foremost is to identify a milling media where the drug has very low solubility. Bead size and concentration of beads versus drug is important. And smaller particles are most successfully produced using smaller beads and higher energies. In some cases, a pre-grinding may be needed with larger beads to make the process more efficient to get to the smaller particle sizes. We spoke a moment ago about the criticality of parameters for successful bead milling. If the drug has some solubility in the media at room temperature or slightly elevated temperature, it will recrystallize onto itself, preventing small particle formulation. This is called Oswald ripening. Viscosity is also critically important. The drug and the beads must be able to flow against and over each other to get efficient grinding. For this reason, surfactants may be used at low levels, but there's a word of caution here because some surfactants may increase the solubility of hydrophobic drugs. So screening for solubility in your grinding media is important at both 25C and elevated temperatures of 37C. It's also common to use particle aggregation inhibitors such as hydroxypropylmethylcellulose and hydroxypropylcellulose, and you'll see these in many formulations. It is often the case that the milling formulation is also the injected formulation. This is to prevent washing and concentration or dilution of the suspension after milling, and can be a very efficient process for making these formulations. Standard formulation design approaches are used to optimize these ratios and excipients. Now let's move on to some examples, excuse me, of micronized drugs. First goal in the form selection is to identify a crystalline solid form that is suitable for micronization and slow dissolution. The target in this case was to convert a daily drug to a monthly drug. The first step is to identify the crystalline form, and this is done by XRD and melting point. And the second step is to test the solubility and then scale up the chemistry and reevaluate these physical properties. Third step is to process the particles by a suitable technique and then confirm that the physical chemical properties are the same, that the X-ray powder diffraction pattern has not changed, the solubility and the melting point are identical as before. Many salt and co-crystal forms were screened for crystallinity and melting point, and then finally screened for solubility if those properties were suitable. Solubility was determined at 25 degrees and 37 degrees, and these two forms were identified for drug 1 and drug 2 after a number of 10 or 20 different forms were tested. The target in this high concentration suspension study was to extend the PK profile out to more than 30 days in rodents and hopefully larger animals. The target particle size for the suspensions is typically below 1 micron for nanoparticles and in the 5 to 10 micron range for microparticles. And this is really designed to enhance injectability and dissolution rate of the drug. In this case, roller ball mill and planetary mixing were chosen after investigating best processes. Typical micronization procedures and parameters are shown in this table. The roller ball mill on the left produced microparticles in the case of these two drugs, and they were operated at a 0.5 gram scale with 10 times the mass of beads in this system. These were 2 millimeter beads, and depending on the drug properties, the time and duration of the milling time was different. The bead sizes were varied and were obviously much smaller for the smaller particle sizes desired in the case of the nanoparticles. In the case of the nanoparticles, it was also necessary to start with larger bead sizes and halfway through the process to add the smaller beads. The suspension obtained was either concentrated or diluted if necessary to obtain the optimum concentration for injectability. This is the final set of conditions. It should be noted that there were many, many conditions explored, including varying excipients, ratios of drug to bead, milling time, and size of beads. Just to give you some idea of the properties of the starting material for drug 1, this is a picture of the crystalline material, which is quite needle-like, and the Horiba laser light scattering particle size distribution on the left shows two modes, one at about 10 microns and one at the larger size of around 100 microns, which is likely the long axis of the needle. It's always important to take a picture using a visual microscope or an SEM to verify and understand particle size distribution data. Drug 1 was produced into microparticles after roller ball milling, and you can see that the particle size was reduced from about 10 microns with some larger particle size down to about 7 microns in this case. So you can see that the larger particles and the long needles were nearly eliminated from this formulation. The light microscopy is consistent with the laser light scattering in this case. The nanoparticles produced for drug 1 in the planetary ball mixer shows particles that are well below 1 micron with a second mode at about 10 microns. And laser light scattering at the top is suggestive of a bimodal distribution, while at the bottom, you see the dynamic light scattering method showing about 2 microns in size. So there's some discrepancy between these two techniques. If you look at the microscopy, you can see that these particles are certainly mostly below 2 micron in size. This is drug 2 starting material, and you can see in this case, there are two closely associated bimodal distributions, one a little bit over 10 microns and a second around 100 microns. Visible light microscopy suggests these particles are in the 10 micron range with another much larger distribution, which may or may not be aggregates or larger particles. The microparticles produced from drug 2 are shown in the image here, the light microscopic image. We were not able to obtain laser light scattering data on this particle because of the color, which interfered with the detector in the laser light scattering system. The nanoparticles produced from drug 2 in the ball mill, in the planetary ball mill, are much closer to 1 micron than 10 microns by visible light microscopy, and the dynamic laser light scattering technique shows about a 1 micron particle size. Again, Horiba laser light scattering analysis was not obtainable due to the color interference. So these are the particle systems that we then formulated using various techniques. The first and foremost was to look at in vitro release testing at 37 degrees in phosphate buffered saline. And we often use initial IVR data for the drug crystals with no processing to understand the baseline. And you can see that drug 1 and drug 2 are fairly close to each other in terms of in vitro release profiles. The manufactured formulations on the right, however, show 3 drug nano and microparticles that are close to each other, with one of the drug nanoparticles having a much higher dissolution rate in the in vitro release system. So note that the 3 lower distributions on the right of the slide have about 5 to 7% release at 4 days. And this is fairly comparable to what was observed in the larger drug crystal particles that were not ground on the left panel at 4 days, showing about 8 to 10% release. The one particle that is quite different is the nanoparticle drug 2. And I think we'll find from looking at the solubility data that the solubility was quite different for that system. So in this table, it can be seen that drug 2 nanoparticles have the highest solubility. We're looking at column 2 in this table. This is an equilibrium solubility for 24 hours in PBS, mimicking the in vitro and in vivo condition. And you can see that drug 1 microparticles, nanoparticles have quite low solubility at 20 micrograms per mil, in the range of interest for this kind of a formulation. Drug 2 microparticles are a little bit high at 60 micrograms per mil, and drug 2 nanoparticles are quite high at 130 micrograms per mil. And that probably explains the difference observed on the prior slide, where drug 2 microparticles are a little bit elevated above both particles for drug 1, and then drug 2 nanoparticles are significantly elevated over all of the others. The in vitro release rates obtained in mg per hour in 2 milliliters of phosphate buffered saline are 0.1 for the first 3 microparticle systems and nanoparticle systems, and 0.3 for the higher soluble system. One of the things that we like to do in estimation of what might be going on in vivo is to approximate the amount of drug, the amount of time required to dissolve the drug dose at the injection site in the human body. And we estimate that the body contains about 6 liters of fluid, which perfuses through the intramuscular tissue in a 24-hour period. So we just scale the amount that's released from the in vitro release data in time up to per day, and then use that to estimate the number of days to release the target dose. You can see that the 2 slowest dissolving systems, drug 1 micro and nanoparticles, we're estimating about 10 days to release the dose. Drug 2 microparticles, we're estimating 8 days, and drug 2 nanoparticles, the much faster releasing system, we're estimating about 2 days. This is a very crude estimate that does not take into account any information on pharmacokinetics, half-life, or clearance of the drug from the injection site. We also evaluate injectability of these micronized drug suspensions. These are quite concentrated suspensions at 200 mg per ml of active drug and a higher solids content of the drug formulation. We typically use an Instron force tester to evaluate the flowability of the suspensions from an injection syringe, and we find that injecting into air is too easy and does not create the kind of back pressure that you see in the in vivo systems. So we use chicken thigh injections as opposed to open air injections to better represent the back pressure that we might observe when we go into the in vivo system and try to inject into the muscle of a rat or a larger animal. 3 of the particles, the top 3 in the row, top 3 rows here, could be prepared at the desired drug concentrations and were injected with reasonable injection forces and break loose forces of less than 5 newtons. The 4th particle suspension was not adequately flowable and could not be tested at the concentrations desired. Significant amount of formulation testing is required to make formulations at 150 to 200 mg per ml in solids content and higher. And the higher you wish to go, the more difficult the formulation challenge. Finally, we come to the pharmacokinetic study, which is outlined here, where a drug solution was injected intramuscularly, and 3 of the particle systems that were obtainable were injected at different doses intramuscularly, all at the concentration of 200 mg per ml. In addition, it's important to evaluate the injection site for tolerability issues and potential injection site reaction when dosing large amounts of drug. In this case, the observable in vivo study went on for 35 days, and during this period, there was no swelling, no limping of the animals, no lame legs, and no pus or exudate evidence at the injection sites. So it was considered to be largely a success. Here's the pharmacokinetics of the formulations on the left. This is just showing 2 of them at the 3 different doses. And what you can see in the upper panel on the left is that the 3 different doses really had very slight differences in their pharmacokinetic areas under the curve. There's an initial release phase and a longer acting second half-life phase of the drug. The nanoparticle suspension on the bottom left, however, showed 3 very distinctly different AUCs and concentration curves for the 3 different doses administered. And the nanoparticles, actually, the green line at the top is much larger area under the curve than the green line above in the microparticle system, suggesting that this is a much better formulation. The microparticles show extended exposure out to about 30 days, the nanoparticle out to about 25 days, and there, in the case of the nanoparticle, there's a clear relation to dose. The first phase of these release profiles is fast, but not quite as fast as the solution formulation shown in the bottom right in the panel. Finally, in summary, it's important to move feasible formulations quickly into pharmacokinetic testing to understand the elimination rate effect on the PK profile from a complex dissolution system like micro or nanoparticles. 2 drug forms were identified and milled by different techniques to both micro and nanoparticles at drug concentrations of 200 mg per mil were found to be flowable with reasonable injection forces tested. The PK profile of these drugs was extended from 3 days in solution out to about 30 days in rats for the suspension formulations. Like most long-acting systems, PK of the specific drug plays a big role in the formulation decision and iterations in the nanoparticle and microparticle systems. Thank you for your attention. I will take questions now.